Hi, welcome to the lecture on environmental adaptation of animals. Now we will consider more detail on the muscle energetics, because we have learned that we had these slow oxidative cells and fast glycolytic cells, and then fast oxidative glycolytic cells, and then there was some variation among these fast cells anyway. But so it must be some differences in the energy metabolism between these oxidative and glycolytic cells. Okay, but let's start on the short review on how to produce energy in the muscle cells. Okay, the easiest way is to just uptake glucose from the circulation and produce ATP with the glycolysis. Okay, we know that it's not very efficient because you can only produce maybe 2 ATP per 1 glucose. So that's why in a typical cells we also have these mitochondria. So we can have this oxidative phosphorylation. And then you can produce maybe 30 ATP per 1 glucose, but also you need then oxygen. And so far we have learned, okay, that limits the size of the cell. So the cell diameter is a little bit more narrow. So the oxygen is easier to pass in the center of the cell. And there's also more circulation around these cells. But also, when we have the anaerobic glycolysis, that's also affecting that, okay, now you are producing lactic acid, and then you must get rid of it. But the good point in these mitochondria that you are not only related only on if you have glucose or not, but you can also use fatty acids, protein, so different kind of energy source for the ATP production. And okay, when you have the ATP and then you are contracting, of course you need for the contraction and all other processes the ATP that will cause that, okay, there's more ADP, but of course you have the creatine phosphate, so these phosphagens to give the phosphate to ADP to produce ATP. So now you have 3 different ways to increase the amount of ATP in the cells. And all these ways are very important in the cells because in the resting cell there's maybe a few millimoles per liter of ATP in the cytosol, and that can sustain contraction only for a few seconds. So any kind of movement, you need the ATP production. And it's not only used for this movement of myosin and actin, but also for the detachment. And then, of course, the structural change of myosin, that is usually the only thing that we remember where the muscle cells are using the ATP. But also we use a lot of ATP for calcium pumping, because if you release calcium inside the cell, then you must pump it back. Okay, we have now at least 3 different ways to produce ATP in the muscle cells, but we have learned from the general biology courses that, okay, the aerobic catabolism is the best way to produce ATP because you can produce maybe 30 ATP per 1 glucose. So why not all the cells are using it? The problem is that on the resting cell, you are using only a few ATP per time, but when you are running as fast as possible, you are using a lot of ATP, and then you must also upregulate the ATP production. And this aerobic catabolism is not very good to increase the ATP production. So with the phosphagens, you can release immediately a lot of ATP, or actually you are increasing the phosphate for the ADP to produce ATP. But anyway, the ATP concentration can be increased very fast. And even in anaerobic glycolytic, you can upregulate these enzymes to produce more ATP quite fast. So with these 2 ways that are not using oxygen, you can increase the ATP synthesis quite fast way, within seconds. But the problem is that they are not very good to sustain ATP concentration because they have very limited speed. With glycolysis, you can produce only 2 ATP per one glucose molecule, and that's not enough for these contraction machines. So that means that then the ATP production will be lower than the consumption, and then the ATP concentration in the cell will decrease. And then you have less ATP in the cells, you have less contraction, the cells are getting weaker, the muscles are getting weaker, you can't run for ages. And of course, in the phosphagens, if you have these creatine phosphate, high number of creatine phosphate, that's a battery. So you release all the phosphor from the battery to produce ATP, but then the batteries are out of charge. Then that's why these phosphagens are only for a very short period. But in comparison, the aerobic catabolism, you can, if you have oxygen available, then you can maintain very high ATP production for ages, actually forever. And this actually causes that because these anaerobic ways are not enough to produce the ATP, then little by little this aerobic catabolism becomes the dominant way. So the acceleration of ATP production in phosphagens and anaerobic glycolysis, they are fast, but this aerobic catabolism, you can speed it up quite slowly. But it doesn't matter because these two others, they can produce so much ATP for the cell. So that's why, anyway, on the long run, you are relying on the aerobic catabolism. And this was something that we already learned when we were discussing on the metabolic rate during the exercises. Okay, if we have now glycolytic cell and oxidative cell, they must have some differences in their pathways. Some mechanisms are operating better in one or another. Well, the glucose uptake is working in quite much in the same rate. They have the same glucose transporter on the membrane to uptake the glucose from the circulation. But these glycolytic cells are more efficient in several steps of the glycolysis, and that's why these arrows are in red. So if you have the same amount of glucose in the cell, these fast glycolytic cells produce a little bit ATP quite fast with their glycolytic enzymes. And these oxidative cells are then producing later more ATP by Krebs cycle and oxidative phosphorylation. But actually these glycolytic cells are using it for different purpose. So instead of turning pyruvate to acetyl coenzyme A, that is over here in the green arrows in these oxidative cells, these glycolytic cells are putting the glycerol 3-phosphate inside the mitochondria to produce dihydroacetone phosphate. So they are just using it for one modulation of one single molecule. Okay, then you have different ways to produce the ATP. But what about then on the lactate? These fast glycolytic cells are having a high concentration of lactate when they are in the work, so they have good ways to get rid of the lactate, so to release the lactate into circulation. But that's interesting that in these low oxidative cells over here in the green, they have now better way to uptake the lactate inside the cell. So if we remember on the morphology of a typical mammalian cell, we have mammalian muscle, we have these different kind of cells just or close each other. So if we have one cell there that is glycolytic, it will produce quite fast energy for the contraction, but it also releases the lactate. And then the next cell, if that's oxidative, that will uptake the same lactate to produce energy with the oxidative phosphorylation. And not only they are uptaking lactate, but then they also have good ways to uptake fatty acids for the energy metabolism. So we can see in this picture that these glycolytic cells and these oxidative cells, one is better in the glycolysis and release of lactate to the environment, and the other one is better in the producing from the pyruvate to carbon dioxide and a lot of ATP, but also not only from pyruvate, you can also use the fatty acids or form lactate to pyruvate. Okay, so there are some fundamental differences in these in the biochemistry of these cells. Okay, what happens then when you have a glycolytic cell and oxidative cell? What happens on the ATP concentration? We learned that, okay, if we don't have the ATP production, the ATP can work for contraction only for a few seconds. Well, with these glycolytic cells, with the glycolytic ATP production, the ATP concentration still drops about 1/3 during the first 10 seconds. And that will also affect then the maximum output of the exercise will drop. So there's less ATP, there's less contraction, there's less movement of the cell, and that's why it's getting the fatigue. So there's less power. And so although during the first 1 or 2 seconds, the cell was using the ATP inside the cell, and then it was using the creatine phosphate battery, and then it was upregulating glycolysis. So it's not enough to maintain the ATP concentration. It's not enough to maintain the power of the skeletal muscle. And when the ATP concentration is decreasing, then of course ADP is increasing. And the ADP in the resting cell is very low, but it rises about 10-fold during the exercise. And even the inorganic phosphate increase about 8-fold during the exercise. And we have learned that, okay, these are enhancing the ATP production. So there were some key enzymes that were upregulated, for example, by ADP. Or if you have ADP, then you are charging ADP to ATP with this phosphagen battery. But that's not going to work for a long time. But anyway, there are dramatic changes in the ATP, ADP, and phosphate in these glycolytic cells. Then, if we have a little bit more oxidative capacity, the ATP drop is much milder, about 1/2 of this fatigue sensitive cell. So then you can use with the same speed the muscle or the same force, the muscles for twice as long. And if you have these fully oxidative cells, the ATP drop is very minimal. There's no change during the first 10 seconds. And even after that, it's only few millimole of the ATP that has dropped. So nothing like the glycolytic cells. So that's why it's better to have a mixture of different cells in different muscles so you can move all the bones. Okay. The problem is that so far we have discussed that, okay, we have fast cells, we have slow cells, we have oxidative cells, and we have glycolytic cells. But the problem is that, okay, we don't have only mice and man, but there are also other kind of animals. And these general rule is that these small animals, they have more oxidative metabolism and more mitochondria. In spite of the abundance of these glycolytic cells in their skeletal muscle. So they have over here in the right hand side there's screen. There's a huge amount of mitochondrial volume. It's much higher than in human, but also there are more 2X or 2B type muscle fibers. Whereas in the humans, we have more type 1 or type 2A cells and less mitochondria per volume. And that's why the comparison is quite drastic. That, okay, in the Screeve's muscle cells, there's maybe 30% of the cell is mitochondria. And in humans, maybe 2 or 5% of the cell volume is mitochondria. And that gives some kind of clear description. For not only that, okay, these small animals have much higher metabolic rate, but also they can't produce so much force, because if 1/3 of your cell volume is mitochondria, there is 1/3 less space for these contractile elements. So in the human cells, although we have oxidative cells, but still if 2% of the cell volume is mitochondria, it means that there is 98% of the cell volume can be packed with these contracting elements. So you can have more force per one cell. Of course, there are not nucleus, etc. But anyway, that's a very large difference in how many contractile elements you can have in a single cell, and how much force you can produce with the same amount of cells. Okay, they have drastic changes in the oxygen use, and they have different kind of fiber types. But because they have different amount of oxygen use, there's also different amount of mitochondrial enzymes. So in these small animals, they have much higher citrate synthase activity. So that's why they can produce more ATP with their mitochondria. And there's also quite large abundance in the fast and slow muscle cells. So if you take histologically or anatomically the same muscle, we have learned that, okay, soleus muscle is the slow one. Still, in the screeve, it's packed with these fast cells. 100% of the cells are fast. Then in the mouse, 60% of the cells are fast. In the rat, a little bit larger animal, 20%, and in a rabbit or a human, etc., 0. So they are all slow cells. So not only we have different type of cells in different kind of tissues, but also how much of these cells in the same tissue exist between large and small animals. That's a huge variation. Okay. Even the slow muscles can be fast. We have already learned previously that, okay, there was a comparison. There was dog and mouse and rat, and they were using different amount of oxygen or having different speed in the contraction. And there's a huge difference in how this myosin ATPase activity is operating in different size of the animals. So in these large animals, the myosin ATPase activity is much lower in these slow oxidative cells. But it's not only related on the size of the animal. So we have athletic animals like horse. It has actually faster type 1 cell than or type 2A than in humans. So it's although the horse is much larger than human. So it's not only linear correlation. But so how it can be done is that, okay, in these small animals, there are some non-conservative substitutions in the fast myosin isoform. So there are some point mutations between the large animals and small animals. And actually, they have similar residues in the slow myosin isoform as we do in the fast myosin isoform. And when we are comparing on the fast cells, the difference is much less dramatic. So in the type 2B fast glycolytic cell, it's about the same speed. In this 2X or even 2A, the change between the small and large animal, there are might be maybe two times higher in the mouse versus in the cow, but nothing like it over here in the slow cells. So the slow cells in the small animals are fast. But it also affects then on the contraction time. So not only these small animals are contracting fast, but they can contract only for a short time. So the contraction time in the small animals lasts only tens of milliseconds. But in the humans, they can last for longer time. Either in fast fatigue cells or fatigue resistant cells or these slow cells. And that's why these fast cells in the fast and small animals, they can't produce high forces because there's no space for these myofibrils in these mitochondrial pacts. And this causes dramatic differences in, for example, in charving muscles in the different animals. So if you have a carnivore that is tearing the prey in small pieces, you must have large cells, large muscles, and you must be able to produce long enough force to get enough power for this mechanism. Or if you are then, even the herbivores, they have quite large muscles in the charving muscles. So it's that's why the small animals and the large animals are even are using different kind of prey or different kind of food. Thank you.